Good morning. We'd like to get started. I'm Barb DuPont. I'm your moderator for this morning. I'm from the Genetics Center, Greenwood Genetics Center. And it is my pleasure to introduce Gavin Arno, also from Greenwood Genetics Center. He is currently the Associate Director of Innovation at the Greenwood Genetics Center and Associate Professor at UCL Institute of Ophthalmology in London. And, not sure I can even read my handwriting on this last one, the Laboratory of Visual Physiology and Ophthalmic Genetics, National Institute of Sensory Organs in Tokyo. He comes to us from the Mayfield's Eye Hospital in London, so we are very pleased to have him, and he'll be presenting to us on the advanced genomic analysis of inherited eye diseases, observations and results from large cohort study and beyond.
Thank you very much. How do I get to?
Oh, sorry.
So thanks, Barb, for the introduction, and thanks for the invitation to speak to you today. So those perceptive ones in the audience can probably tell that I'm not local. Yes, I came to the south from the United Kingdom just over a year ago, and I'll be talking about some of the work from my lab over the past decade or so, working on large cohorts for retinal dystrophy patients in the United Kingdom, and how that work has now continued at GGC. So we will cover a few topics. We're going to talk about the large-scale studies and next-generation sequencing projects for retinal dystrophies in the UK. Moving on to discussing the missing heritability that still is found in genetic testing for inherited retinal dystrophy. And a few of the explanations for that missing heritability. So we'll talk about hypomorphic alleles in otherwise syndromic disease genes, non-coding variant analysis that we are able to perform better and better as time goes by and the technology develops. And then finally, the application of third-generation sequencing or long-read sequencing and the analysis of splice variants with that we perform in our lab, and for regions of the genome and genes that are intractable to short-read or NGS sequencing. So we'll start off talking a little bit about the tissue that we're interested in. And I think it's important to point out now, before you all get too bored of the retina, that I think these lessons and these things that we learn from studying retinal dystrophy are applicable across broader Mendelian and heritable disease. My screen's just gone black. Oh, here we go. Okay. This one is flashing a bit. So the retina, the cells that make up the neural retina at the back of the eye are pictured here. So the light comes in and passes through several layers of cells until it reaches the photoreceptors, where the photon of light is converted into an electrical signal that's then transmitted to the brain. And the image in the corner there is a picture of a normal fundus, a normal picture of the back of the eye in any one of us, general population. And this is what that image looks like in the commonest retinal dystrophy that we see in genetics clinics. This is retinitis pigmentosa, which is a disease which originates in the rod photoreceptors. So it's rod photoreceptor degeneration. And what you are seeing here, if I can work the pointer here, these are called bony spicules. These are the typical pattern of degeneration that we see in retinitis pigmentosa. And a couple of other features that we see typically are the thinned vessels here and the pale optic nerve head as well. So we can image this with different kinds of light and different kinds of imaging platforms, and this is fundus autofluorescence imaging, which gives us a picture of the degeneration of the RPE cells, the cells underneath the photoreceptors that support them, and are degenerating as a consequence of the receptor cell loss. And then finally, this is OCT imaging, where we can look at a cut through the retina layers, and on this image we can see the loss of this cell layer here. This is the inner outer segment layer here. So we do have the ability then for fairly deep phenotyping in retinal dystrophies, and this contributes, I think, to making it a good model for studying genetic disease. There are lots of other factors for that. It's relatively common as a rare disease. It has this huge allelic, genetic and phenotypic heterogeneity. There are over 300 genes implicated in retinal dystrophy to date. Clinical investigations can be very specific and very sensitive. We have the ability to do this deep phenotyping, and the patients are able to tell a very good biography of their disease. The patient's story is often very informative as to what cells are affected on the back of the retina. And so in the UK, we kind of led the world in application of whole genome sequencing for rare disease in the clinical or in the healthcare setting. And the first study of its type was the NIHR Bioresource, which is the National Institute of Health Research, the rare disease study, which spanned a few years, 2014 to 2017. And we played a fairly significant role in that. We performed the analysis for the retinal dystrophy patients within that project and published those findings in 2017 in the American Journal of Human Genetics. And that kind of led on to the Genomics England 100,000 Genomes project, which was kind of born out of the early results of that study, which included 80,000 individuals across rare disease and rare cancers. That's why it's 100,000 genomes, but 80,000 individuals, because the cancer patients got 2 genomes performed in those. And the pilot study of that, the findings were published in 2021 in the New England Journal of Medicine. Both these studies were performed using Illumina short read sequencing, PCR-free short reads, 100 to 150 base paired end reads. And so we'll talk a little bit more about these projects. So the NIHR Rare Disease Project included 13,000 individuals, 700 patients with retinal dystrophy recruited from our hospital, Moorfields Eye Hospital in London. And we started that project in 2013, sequencing. It actually began as an exome sequencing project, but after 6 months or so, it switched over to whole genome because it became actually economically feasible to be able to do that instead of exome sequencing. So the first group of patients actually got 2 tests done. They got an exome followed by a genome, which gave us the ability to compare the analysis of those 2 data sets as well. And so the basic findings of the retinal dystrophy part of that project was that we identified a molecular cause of disease in 56% of the patients that we analysed. And this was based on coding variant analysis, so focusing only on coding variants in a curated gene panel. And these are some statistics from that project, the proportion of solved cases per phenotype and ethnicity here. And what's important to note is that we're still analysing this data, and we're probably closer to around about an 80% molecular diagnosis rate now in 2025, and still making discoveries today. And then the 100,000 Genomes project, which came a little later, was focused on actually trying to sequence families and trios rather than singletons, and studying rare diseases that are non-monogenic, so diseases where there is not only one gene that causes them. Recruitment across the NHS England didn't include patients from Scotland, or Wales, or Northern Ireland. And this data that's generated from this project is accessible to researchers anywhere in the world. What's needed to be able to do that is an institution-level agreement with Genomic England, and then that data becomes accessible for research. There are currently around 4,000 researchers analysing that data. Several groups in the United States are looking at that data, and they are divided into these 8 research communities. And so for ophthalmology then, within that project, roughly 10% of the rare disease recruits were patients with an ophthalmic disease. About half of those came from our hospital, Moorfields, and so we did the analysis for the 100,000 Genomes project and contributed to that pilot study. All of the patients recruited to the study had this clinical interpretation pipeline, which was performed by the clinical labs in the UK. And for retinal dystrophy, this focused on 300 or so genes, which is in-house curated by Genomics England, but also on this platform called Panelab, which is kind of a crowd-sourcing for curation. So people, experts, researchers, clinical scientists and so on can register, log in and help with the curation of genes, add new genes as they're discovered, and that will eventually filter through to the clinical pipeline as well. And so again, this pipeline is really focused on known variants, so known damaging variants in known genes, or likely damaging, likely pathogenic variants identified within those genes. And that's what the patients got fed back to them on their clinical reports. Anything outside of that was research findings, and those were the ones that were made by all of those researchers looking at that data, including our lab. And so the pilot study published in 2021 identified an overall diagnostic yield across all phenotype areas of 25%, and this includes lots of very complex diseases and difficult to solve cases, obviously. Ophthalmology, we did a little better, very similar to what we found in the NIHR study with a diagnostic yield of 55%. And again, this is increasing all the time as we make discoveries and feed this back into the study. And those patients actually get those results as they're published. They will have an updated report fed back to them as well. And then, so in our lab, we're interested in what we're missing from this analysis. So many of the families recruited to these 2 studies remain molecularly undiagnosed, and we think there are lots of reasons for this. There are still new genes remaining to be discovered, hypomorphic alleles, which is something that we'll talk about in a little bit more detail. So these are milder presentations for syndromic disease genes, for example. New genotype-phenotype associations, so unexpected findings that we haven't really predicted before. Non-exonic variants, so non-coding variants, upstream, regulatory, deep intronic, those sorts of things. Structural variants, which I haven't mentioned so far, but these were not very well studied and not very well characterized by the clinical pipeline for both of those studies. And then some of the more difficult to characterize findings. Of course, there would be things like mosaicism, which is not well picked up by whole genome sequencing analysis. Non-Mendelian inheritance, which is an area of interest for lots of researchers and lots of groups, where we have more than one gene contributing to the disease, and non-genetic disease, of course. And so part of this is down to the technology, the challenges of short read sequencing. So these short reads, 100 or 150 base pairs generally, are mapped back to the reference genome, and that's complicated for many regions of the genome. The context of those reads is not present. We don't know which allele they come from, for example. We don't know where they map if they are a non-unique sequence. So there are technological reasons why we may miss diagnoses and miss variants. And partly that contributes to undetectable variants, and there are things that we have discovered in our lab and other labs working with us, things like the North Carolina macular dystrophy, which is caused by a large rearrangement, a 40 kb rearrangement in a gene desert on chromosome 5, one of the causes of that disease, which is obviously difficult to detect, first of all, but difficult to interpret as well. Things like transposable elements, which are very complicated to detect with short-read sequencing. Low complexity regions, and there are a couple of examples of that for retinal dystrophy genetics, and I'm sure people in the audience have got their very well-known genes or very complicated regions of the genome that they know are very difficult to sequence with short-read sequencing. And finally, I'll just mention the last thing on this list, these uncertain genotypes. So this, I think, contributes greatly to patients receiving an uncertain clinical report, and this is due to the fact that we may identify two variants in a gene that is implicated in the disease we are studying, but without the ability to segregate, without access to families, it's impossible to get that report to a confirmed molecular diagnosis, and that's difficult for many patients that we see in retina clinics. So we'll just go through a few of these things then. So first of all, gene discoveries, novel genes that are contributing to diseases. These are some of the genes that were discovered from these two studies in our lab. There are many, many more genes that have been discovered that we've contributed to, but these are the ones that we found in our lab from these two studies. But we'll move on to hypomorphic alleles of syndromic disease genes. That's an emerging phenomenon, I think, in the post-genome era, and something that we're seeing more and more of. So if we look at some Omian statistics, we can see there are over 7,000 phenotypes described in Omian, but only around about 5,000 genes described in Omian. So many of those genes, I think it's somewhere around about a quarter of those genes have two different phenotypes listed in Omian. So this is something that's fairly well characterized and fairly well known. And so in the post-genome era then, these new and unexpected gene disease associations are indeed an emerging phenomenon. And in the retinal dystrophy world, we are able to discover, I guess, or characterize or describe these with more confidence once we've reviewed and exclude all of the known retinal dystrophy genes from our genome analysis. And often these findings are recurrent, so these large-scale projects enable us to detect and find recurrent mutations across several patients and families in these studies. Predominantly they are missense, and they often have a higher minor allele frequency than may be expected for a severe allele in that syndromic disease gene. And so this unbiased approach enables us to discover these large-scale projects enable us to find them, but it's the advances in variant interrogation and analysis of variants that really enables us to be able to characterize and drill down into those. And so for retinal dystrophy, this is a graphic from some colleagues in Europe, the ERDC, European Retinal Dystrophy Consortium, which describes the non-syndromic and the syndromic genes that are responsible for retinal dystrophies. And so far, there are 56 genes that appear in both of those halves of that slide. I won't ask you to pick some out for me. But broadly, they span several disease areas. So ciliopathies is a recurrent theme for this. Mitochondrial disease, metabolic diseases. The mechanisms are often not well understood, but maybe some of the reasons that we can start thinking about for this is that the photoreceptors have this very high metabolic demand this very high energy demand, and that may make them more susceptible to metabolic disease, to the first tissue affected by a metabolic disease, possibly. And then there may be alternate or secondary functions for some of these proteins in the retina or retina-specific isoforms, so particular exons of these genes that are only expressed in photoreceptors or retinal tissue. And so one example that we have discovered and characterized in our lab a few years ago is coenzyme Q10 deficiency. So typically this category of diseases caused by genes in the pathway. I think there are 13 different genes contributing to the coenzyme Q10 biosynthesis pathway. These typically lead to neurological disorders, macrocephaly, cardiovascular disease affecting multi-organ systems, and are consequent on these decreased levels of coenzyme Q10. And so a few years ago we found these variants were accumulating in our unsolved non-syndromic retinal dystrophy patient population. And this paper came out in 2022 from my lab. It's my former PhD student, Neringa, who did this work. And these were the families that we identified, that we studied as part of this project. So these are, the fairly typical retinal dystrophy images that we're now all very familiar with, that were observed in these patients. These patients had none or very, very mild extraocular features, so no real suggestion of a syndromic disease gene. And we found 4 genes in the pathway with biallelic variants in this group of patients. And so some of the notable findings from that then are represented here. So PDSS1, we identified 6 families with variants in this gene, so 9 variants across these 6 families. None of them were biallelic nulls, which is perhaps what we would have expected, given that a biallelic null in this gene would lead to a severe syndromic disease. And this one particular variant, this lysine 197 variant, was recurrent across 3 of the families. And so this variant's got a fairly high allele frequency in nomads over here, and has been previously seen and previously reported in ClinVar as a likely benign variant. And so here we've seen it in 3 families, each time in trans with a severe loss of function allele, and so we believe this is a pathogenic hypermorphic allele. A little bit of statistics we did on this, found an odds ratio with p-value here. This is a significant p-value, an odds ratio of 108 for this type of genotype. And so COC2, again, recurrent missense variant seen in the 3 families that we identified. Again, previously reported in primary coenzyme Q10 deficiency this time. And COC5, which in OMIM only has a single entry with a homozygous 9 kb duplication spanning that gene. And here we found this -7 splice region variant in trans with different loss of function alleles. And we did some splice analysis, some RNA analysis on that, and demonstrated that it has an incomplete effect on splicing. So it affects splicing, but it's leaky. So there's our mechanism for a milder effect, I think. And so more recently, this paper came out earlier this year, I think, which found, again, biallelic variants in the same pathway, a different gene in the same pathway, again, in non-syndromic retinopathy. So this kind of confirms our findings as well. So we will move on then to non-coding variant analysis. And this, I think, is a huge area for discovery in rare disease genetics. And so this paper we wrote a few years ago And it's kind of focused on picking out the patients, the individuals where we have a clue as to what gene we are looking at. And that clue may be a genetic clue. They may carry a heterozygous pathogenic allele in a gene that fits their recessive disease phenotype. Or they may have a very characteristic phenotype that points to a single gene or a very small number of genes that we can really analyse in great detail and look at non-coding variants and indeed structural variants and really drill down into that data. And I think this is kind of the starting point for non-coding variant analysis, because it's very difficult to do this on a genome-wide scale, because there are millions of variants in the non-coding region of the gene, and identifying, characterizing which of those is damaging a protein product is very, very complicated. So we focused on a cohort of patients where we had a pretty good idea of which gene we should be looking at, and identified a pathogenic genotype in all of those cases. And this is one example of that here. This is a lady who presented with non-syndromic retinopathy, and she was found to carry this stop gain in the BBS10 gene. Now, BBS10 is obviously Bardet-Biedl syndrome, but also causes non-syndromic retinopathy as well. And we looked in more detail at that data and found this upstream variant was really the only candidate variant within that gene that we could identify. And so we chose to do some luciferase assay analysis on this. We generated the mutant, we generated different deletion constructs, and demonstrated that this variant, the second construct here is the mutation, reduces but does not abolish transcription of BBS10. So again, this is perhaps a mechanism of how this is a milder, hypomorphic allele for the gene. And then we'll talk more about splicing variants. So this is something that we are particularly interested now in my lab. And of course, these can be coding. They can be close to an exon, or they can be deep intronic. Now, I mentioned the difficulty of being able to interpret non-coding variants, and so I think the best tool that we have now for predicting splice effects is Splice AI. But variants will often or usually need functional investigation to really prove that they are affecting splicing, to confirm a pathogenic effect. And so for this, we need to do RNA analysis. For retinal dystrophy genes, they are usually limited to being expressed only in retina tissue, so these may not be detectable from blood RNA, and the effects can be complex, they can be multiple. Really, mini genes and other methods are a bit too complicated for clinical labs to be able to take on routinely for individual patients, so we need a new way of being able to analyze these. And so in our lab we turn to nanopore sequencing for this, which enables us to detect complex and low-level splice effects, and enables us to detect splice effects from genes that are not expressed or have very, very low expression in blood. And this was one of the first genes we tried this out on, the TOLP1 gene, which the plot here, the expression plot shows that it's retina specific. And this variant was actually part of the 1998 discovery paper, this -6 splice region variant. And in that paper they state that they couldn't confirm this was pathogenic because they couldn't detect the transcript in blood. So now, 27 years later, we can do Splice AI analysis and show that there is a fairly convincing effect on splicing for this variant. And we can use nanopore sequencing on a blood sample from a patient who carries this mutation and demonstrate that there is indeed a very definite splice effect, and it's complete, and it abolishes the canonical acceptor site and forces the spliceosome to use this cryptic intronic splice site, leading to loss of function of the protein. So another example of this type of analysis, the CEP290 gene, again we can see here that there is pretty much no expression in blood. And this missense mutation this time was identified in the homozygous state in a patient with early onset retinal dystrophy, but it has been interpreted as being likely benign or at best a variant of uncertain significance. But when we do splice AI analysis, there's an even more convincing splice alteration for this variant. And so we did RT-PCR and nanopore sequencing to test the effect of this on a blood sample, and again found that it introduces this cryptic splice site within the exon, so again leading to loss of function of the protein. And so finally we will talk about, just to say before I move on, we've got many examples of this sort of analysis now, and we're collaborating with groups not only in the retinal dystrophy world, but outside of that. We'll accept any challenge to be able to characterize a splice alteration for your harvest gene that you have. So finally, then we'll talk about low complexity regions and the analysis that we're able to perform now with long and ultra long read sequencing on these difficult to solve cases. So this is one of my favourite figures from a paper in 2019, which illustrates the spectrum of diseases and some of the genes that are implicated in genetic disease that are very difficult or impossible to sequence with NGS analysis. And there are a couple of well-known retinal dystrophy genes in that graphic. One of which is RPGR. So RPGR retinopathy is the commonest cause of X-linked retinopathy. It accounts for roughly 80% of X-linked retinopathy in males in clinic. I think somewhere in the region of about 20% of patients seen in the Moorfields clinics have RPGR disease. Molecular diagnosis is challenging. Most of the mutations are found in this final exon. I'll just skip forward a second. This final exon here, which has this very poor coverage region, which is the red sequence, which is a repeat region. The exon is about 3 kb, and this region is somewhere around about half of that exon. Now, most of the mutations in the gene occur within that repeat region. So this makes molecular diagnosis very challenging. In the UK, it's done by a separate test, which is based on long-range PCR and Sanger sequencing. Other labs around the world will put more probes into that region with exome sequencing or targeted gene panels. Another region is the opsin array, which is an array of repeat genes which govern colour vision, and severe disruption of this region causes X-linked cone dysfunction syndrome. And this is a complete black hole for short read sequencing. So again, we turn to Oxford nanopore long read sequencing to enable us to read through these complex regions. And essentially what we are doing is making the reads longer, which makes the mapping simpler. That's really all we're doing here. And so we now use a method called adaptive sampling, which is a really cool method, which it's a bioinformatic method that enables us to control the individual pores of the flow cell in real time while it's sequencing. So we can reverse the voltage for an individual pore and push a strand of DNA back out of that pore and stop sequencing it if we're not interested in it. And so this enables us to enrich our experiment for regions of interest. And we use this in our lab to target a panel of retinal dystrophy genes and analyze these with this new technology and compare that data then to short read sequencing data. And these are our basic results from this. We get complete coverage of all of our genes of interest. We are able to phase variants across every single one of those genes. We're able to cover all of those complex and difficult to sequence regions. And so the RPGR poor coverage region, which I showed just now, is now very well covered and very evenly covered with long-read sequencing. And the opsin array, which is kind of the holy grail for sequencing, low complexity region sequencing is well covered, and especially nice when we map it with the T2T reference genome. And I better rush through these last couple of slides. So phasing through large genes. So all of the gene panel, we're able to phase every single variant across all of the gene panel, including the largest retinal dystrophy gene, which is EYS, which is 2 megabases on chromosome 6. We can phase variants at either end of that gene from a singleton. We don't need parents for phasing anymore. And so we've done this on a series of clinical cases now. Patients with opsin defects, some positive controls from more fields, some suspected ABCA4 retinopathy cases, and some patients recruited at GGC with a suspected IRB who had concurrent clinical testing as well. And the results, the headline result for that is that we're able to characterize the pathogenic genotype in all of those individuals. And so I'll just wrap up then. In summary then, whole genome sequencing does enable us to characterize the vast variant landscape across the genome, somewhere around about 95% of the genome. There are challenges remaining for retinal dystrophy genetics and of course all Mendelian disease genetics. And we've talked about some of the reasons for that missing heritability that we've been able to characterize in our lab, and the limitations of the short-read sequencing that we do now. So new technology can help us overcome some of these limitations. Ultra-long-read sequencing in particular enables this distant variant phasing and the characterization of low complexity regions. And so I'll finish there with my acknowledgements, and if there's time for any questions, I'm happy to take them. Thank you very much.
Questions for Dr. Arno? Of course.
So, fascinating talk, an amazing amount of data. Sort of an unfair question, okay? Sorry, I'll preface it. But I wonder if all of the data that you're collecting at the DNA level allows you to begin to put together, how does a retina come into being in embryo or in fetal development? It's very satisfying to find DNA causes for disease, and it would be really satisfying to be able to then start to put some of these genes together, gene functions rather, together in terms of, how does this organ actually work? Mm-hmm. And I'm thinking a little bit about the cobalamin C phenotype, which has both a variable retinopathy in some patients and others don't have any, as well as variable microcephaly, and I'm not sure what my question is, but
So, okay, well, I'll answer.
I'm just thinking about functional.
Yeah.
How do we get to a functional understanding of the many DNA variants you've described? I think one thing that's difficult about studying retinal development is that the difficulty that we have with generating things like RNA-seq data from retinal tissue, particularly from patients with disease, is virtually impossible. We rely on enucleated or post-mortem
tissue to be able to study transcriptomics. What we're hoping to be able to do now is, so one thing I didn't really mention is the fact that long-read sequencing also generates methylation data from the DNA sequencing. And so we have a project starting up now where we're going to start to perform these experiments on normal retina tissue and start to look at the methylation data, start to link that with transcriptomics data, RNA-seq data, and start to see how the methylation of that specific tissue relates to the expression of the genes and proteins. But this is adult tissue. Getting that from developing retina is even more complicated. So we generally have to rely on animal models to study the function of genes in development, but then again, most animal models don't have a retina that replicates the structure and function of the human retina. So there are huge gaps in our knowledge still.
I assume it's not possible to culture retina cells?
So it is. So there are lots of groups generating eye cups from iPS cell lines from patients. So there's lots of work in that area. Most of those studies are kind of focused on studying gene therapies and early tests for the effect of gene therapies. So there are groups interested in retina development, but those studies are still a way out, I think.
Thank you very much.
Okay, thank you.